All right, welcome guys to the first day of online school. Make sure you have the skeleton PowerPoint open so that you can take notes on it because that is your assignment. That is going to be due tomorrow by 8 a.m. That's Tuesday the 17th by 8 a.m. So make sure you've got it in there. Also pay attention to your quiz date in OneNote. That is also going to be Tuesday the 17th. Socrative will be open for your quiz from 11 a.m. to 12 p.m. and it will cut off right at 12 p.m. So make sure you get it done in time, otherwise it will be a 0. The class code, as a reminder, is Kalen4870, but that's also in the directions in OneNote. Also, I hope you've watched the introduction video because that is also part of your assignment today. So make sure you do that. Otherwise, that'll be a 0. So knock them both out and you'll be just fine. We're going to start with the cell cycle today. So up to this point, we've already talked about what is cell division used for. In multicellular organisms, it is cell division for growth, maintenance, and repair. However, in single-celled organisms, it's just to reproduce. So that's the main function there. Then we talked about the cell's DNA and how it makes up the genome. Just as a reminder, the genome is the complete set of genes or genetic material present in a cell or organism. So basically you take a DNA sample from an organism and you map out its entire genome. And it gives you a really good understanding of how you could, from a genetic standpoint, build an organism. Then we talked about how we make chromosomes. We talked about the number of chromosomes and how they vary among species, but they're consistent within a species. So we talked about cats and dogs have vastly different numbers of chromosomes. Dogs with 78, cats with 38. However, all dogs have 78 and all cats have 38. So they're consistent within a species, but they vary among species. Then we talked about the difference between a body cell and a sex cell. And again, what the difference between a diploid and a haploid cell was. A diploid, they have 2 matched sets of chromosomes. Wherein a haploid, they have half the number of chromosomes. And we talked about why they have to have half the number of chromosomes. So if you had 2 diploid gametes, you would have a cell that had way too much DNA. So you have to have an egg and a sperm, both with half, so that when they come together, you have a cell that has the correct number of chromosomes. We talked about a karyotype and how certain chromosomes are homologous and certain chromosomes are heterologous, with your X and Y being heterologous and your all your other chromosomes being homologous. And then we talked about females, XX, males, XY. Make sure you remember this from class. That was all the stuff we did with chromosomes and karyotypes and whatnot. We talked about how you can condense DNA. Make sure you know histone proteins and nucleosomes and chromatin and finally chromosomes. All important stuff. Today, however, we're going to start something new. We're going to look at the cell cycle. And all the cell cycle really is an ordered series of events in the life of a cell. So what you see below there in the diagram is the life of a cell. And it's broken into 2 very distinct phases. Those 2 phases, you can ignore this diagram or this picture, it's not for you all. The cell cycle has 2 major phases, interphase and the mitotic phase. And you can see the interphase there because it's all in blue. All right, it's a time for normal growth and preparation for cell division. So if I said, does the cell spend the majority of its life in interphase or the mitotic phase? You would say interphase. The interphase is comprised of a G1, an S phase, and a G2. The mitotic phase is the phase of mitosis in which the DNA and cytoplasm are split and the cell physically divides. The interphase is really important. However, the mitotic phase is where all the action really happens. So that's what we're going to go through today. So let's get into it. You do notice the gap 0 phase. We'll talk about that later on when we talk about what regulates cell division. But for now, you can just ignore that G0 phase and just focus on G1, S phase, and G2. So interphase. G1 phase, or the first gap phase. In this phase, change is not evident. It's very similar to a gap. But the cell is biochemically active, and biochemically active sounds fancy and complicated. It really just means that the cell is growing, so it's a little bit of growth. It's not, it sounds more complicated when you say biochemically active, but it's just growing. Then it moves into the S phase, and in the S phase is when the DNA synthesis occurs. So our last chapter test covered, the structure and function of DNA. It covered DNA replication. This is where DNA replication occurs. So all of that stuff we learned just a few weeks ago with the Okazaki fragments and the leading and lagging strand and why are we making a copy of our DNA, this is when that happens. It happens during the S phase of interphase. So S phase, synthesis, is synthesis meaning to build. It all makes sense. And then you have the G2 phase, or the second gap phase. Within this phase, you start to replenish energy. So DNA synthesis takes a considerable amount of energy. So it would have been hard for the cell to go directly from the S phase into the mitotic phase. So you've got this gap 2 phase where the cell can replenish its energy and get ready to divide. Your organelles reproduce, because if you're going to create 2 cells from one, you obviously have to have much more organelles present in order for the cell to live. And then lastly, you have something called a cytoskeleton breaking down. Don't worry too much about that. Really just focus on energy being replenished and then producing more organelles. And then also in G2, there is some cell growth as well. So interphase is really easy to remember based on how they're named. G1 phase, G2 phase, growth, GG. So that's one way you can remember it. And then S phase, synthesis, new DNA. So you're copying the DNA. So I really don't anticipate it being that hard to learn interphase based on the names. Not a super exciting phase of the cell's life. The S phase obviously has the most going on. And you should know a little bit about the S phase because we've talked about DNA replication. All right, the big stage, the mitotic phase. There's several steps to the mitotic phase, and this is what we're going to spend the bulk of this video on. And if you're 6th period watching this, you've seen this before. Go through it again. I know you don't remember it because we only did it briefly. If you're 4th period, this is the first time you're seeing it, so pay close attention because it can get a little complicated. All right. First step in the mitotic phase, because there are 2 words for this. I'm fine with you knowing either one. You have karyokinesis, and it's also called mitosis. You can call it mitosis. I'm putting the big word in there because I want you to just be familiar with it in case you ever hear it. But I'm perfectly okay with you calling it mitosis. You can even call it nuclear division if you want. I'm fine with all of that. But that's the first step. The second portion of the mitotic phase is called cytokinesis. And cytokinesis is when the cytoplasmic components physically separate into 2 daughter cells. It's the actual splitting of one cell into 2. And a lot of people think that cytokinesis is part of the mitotic phase. It is not. It is its own separate phase that occurs after the mitotic phase. So please don't think that cytokinesis is part of mitosis. It is after mitosis. Okay? All right. Let's go ahead and jump in. We're going to start with mitosis. Here is an overview of mitosis. The thing I will say is don't worry about prometaphase. I'm not going to make you know prometaphase. I'm only concerned with you knowing prophase, metaphase, anaphase, and telophase. Those are the parts of mitosis. Again, do not worry about prometaphase. But we are going to look at each phase, what goes on during that phase, what the cell looks like in that phase. And unfortunately, with us being out of school, we can't get in the microscope lab. You would have been able to look at microscope slides and see each one of these phases that had happened in actual living cells. We would have used onion root tip or whitefish cells, which is pretty cool. So again, blame the coronavirus for that. But let's jump in. You're going to want to remember the order that these things happen in, and it's really easy because it's PMAT. PMAT, prophase, metaphase, anaphase, telophase. It's really hard to forget if you just remember PMAT. You don't have to worry about cytokinesis because it's not part of mitosis. All right, mitosis begins with prophase. So one of the first thing that has to happen is the nuclear envelope breaks down. And if you think back to what a nuclear envelope is, it's that membrane around the nucleus. If you're going to get the DNA out so you can form two new nucleus, you have to first break down that nuclear envelope. So that's the first thing that happens. Second thing that happens in prophase is your membranous organelles. Those are just organelles with their own membrane. So examples are the Golgi apparatus, the endoplasmic reticulum, the mitochondria. All of these different organelles move towards the edges of the cell. Now I'm going to come back to this slide, but I want you to see what this looks like. Well, that's what I want to go backwards. You can see in the nuclear envelope, you can see the little the gaps that are starting to form in it, and then you're also seeing how all of the organelles have moved to opposite sides. The nucleolus disappears, and then you have what are called centrosomes begin to move to poles. If we go back to this slide here, the things that look, they have the orange in it, and it looks like they have little spider webs coming out. Those are called centrosomes. So make sure you know what those are. We're going to go through them in just a second, but I wanted you to see them. They begin to move to poles. If you're confused about poles, think about the North and South Pole, right? They're opposite ends of the Earth. The poles that I'm referring to are the opposite ends of the cell. So that's what I'm referring to when I talk about poles. But these centrosomes create what's called a spindle fiber. And these are composed of microtubules, which are proteins that provide framework to move structures within a cell. So the little spiderweb things coming out of the centrosomes in this picture are called the spindle form. And those are going to play a really important role in pulling chromosomes apart. Okay, that is prophase. So that is the first step in mitosis. The next step is going to be metaphase. Remember, you don't have to know prometaphase. Can't stress that enough. Metaphase. The two things that happen in metaphase, your chromosomes are going to line up along a metaphase plate. The metaphase plate is not an actual thing that forms. It is a location. So you can see how the chromosomes are lined up straight down the center of the cell. That is what they're referring to when they call it the metaphase plate. You'll also hear it called the equatorial plate for the equator. It doesn't really matter. You can just call it the center of the cell. But chromosomes line up along the center of the cell. And then sister chromatids, which if you're confused by that, if we go all the way back to this slide, it shows you what sister chromatids are. So if you're confused about that, look at this slide for a few minutes, and that should help you. But sister chromatids remain attached by cohesion proteins. At this point, there is a protein in the center of those two sister chromatids that are holding them together. They will not hold it together for much longer, though. Because in anaphase, you separate those sister chromatids in opposite directions towards the centrosomes. So these microtubules, which are your spindle fibers, your blue lines coming out, attach to those chromosomes. And those centrosomes pull those sister chromatids apart. And you can see how those sister chromatids migrate to opposite ends of the cell. This is going to allow both new cells to have the same amount of DNA, the correct number of DNA. So the cell is also going to elongate. You can kind of see how it's going to form this football-like shape. And that's going to allow enough size for both new daughter cells. So when this cell splits, forms two new cells, you're going to have ample space for both of those cells. All right, that's anaphase. Lastly, we have telophase. And in telophase, the chromosomes have reached opposite poles or opposite sides of the cell. The DNA begins to decondense because it doesn't have to be as condensed as it does during replication or mitosis. You're starting to see a new nuclear envelope around both new sets of chromosomes. You're seeing nucleosomes appear within the nucleary. You're seeing all of the stuff that's necessary to form two nuclei for two new cells. And that's going to occur in telophase. You're also seeing the organelles begin to shift away from the poles and into the kind of fill the space around each new nucleus. So that's the 4 steps of mitosis. The last thing we're going to talk about in the cell cycle is cytokinesis, because without cytokinesis you wouldn't have two new cells. And they're different between plant cells and animal cells. So in animal cells, you're going to form something called a cleavage furrow. And you can see that it's kind of, they've used it with red X's there in this diagram. Those are not chromosomes. That is just where they indicated where the cleavage furrow is. And it will actually pinch off until you formed two new cells. This is different than plant cells. In plant cells, you form something called a cell plate. And this cell plate will eventually turn into a cell wall, and that will physically separate the two daughter cells. What you'll notice is there's some space between animal cells. You don't have that much space, if any, in between plant cells. And that's because you're using a cell plate versus a cleavage furrow. So make sure you can tell the difference between cytokinesis in plants and cytokinesis in animals. The very last slide, pretty short video. We're going to spend literally one slide talking about prokaryotic cell division. So prokaryotes, like bacteria, propagate, and all propagate means is to reproduce. So you could replace that with reproduce. So prokaryotes, such as bacteria, reproduce by binary fission. And this binary fission is the only way they produce new individuals. So you can see the 4 steps of binary fission below. It replicates the DNA. The cell grows a little bit. The DNA moves to opposite sides, and the cells split. So it's kind of a condensed or smaller version of mitosis. They are way less complicated, but because if you think about a prokaryotic cell, they don't have all of their own membrane-bound organelles. So they don't have to go through as much of a complicated process as eukaryotic cells do. That's it for today. It was a quick one. I wasn't going to overload you with too much stuff with it being day 1. Again, do not forget about your quiz on Tuesday. Please, please, please make sure you fill out the skeleton PowerPoint with your notes. Turn that in before 8 a.m. on 3/17, which is Tuesday. Get it done, and have a great rest of your day.